So this is just one sample of the kind of analysis that we're doing. This is a comparison of the microbiome of infants who are fed breast milk versus formula. And this is really just asking the question of who is there, what is the microbial communities that are there. So each bar represents an individual infant, and each colored portion of the bar represents a different bacterial type. The length of the bar shows what percentage of the total bacteria in the sample this bacterial type was made up of. And the purple, just highlighting, is what we would call the commensal kind of bacteria, the good bacteria. The infants were then divided whether they received breast milk or whether they received formula. And you can see that infants that receive breast milk have a much higher percentage of commensal bacteria compared to infants who received formula. So that's just one sample of the piece of information. On the poster is additional information addressing the question not only of who is there, but what are the metabolic processes that are there, and additional information on how the infants track over time more closely within twin pairs than between twin pairs. So in summary, this is a very unique, informative data set. It's been collected prospectively over time with an in-depth correlation to diet, environmental exposure, medical history, genetics. And it's already being used as a worldwide reference for population comparison. Our collaborator, Dr. Gordon, in the Genome Center, is looking at a variety of different infant populations really around the world, looking at issues around malnutrition as well as overnutrition, and using our CDI samples for those comparisons, and we have a paper coming out shortly in that regard. Our early study findings indicate, based on what we saw today, that diet is critical to the assembly of the microbial population. Microbial communities track more closely within families. And importantly, we've now collected and archived a unique sample cohort that we can really use into perpetuity. And hopefully we won't have to send out the poopmobile anymore after we're done with this study. Our collaborations are really just beginning. One of the exciting parts of this CDI study is the support and collaboration that it's built among other investigators. The CDI samples can be used to benchmark against the premature population. We're looking at problems that our preterm infants have related to the microbiome. Necrotizing enterocolitis is a very significant cause of morbidity within that population. That's a local effect of the microbiome, but then also looking at systemic effects. The chronic lung disease that many of our babies can develop is a chronic inflammatory response as well. I've alluded to the fact that we have a close relationship with Dr. Dantes over in the Genome Center, looking at the role of antibiotic resistance and using these CDI samples to track the development of antibiotic resistance and the gut as a reservoir for those antibiotic resistant genes. And then finally, a continued collaboration with Dr. Gordon, looking at questions we talked already about overnutrition and obesity, but also undernutrition and malnutrition.
Art, can I just make a
Yes.
comment here? For those of you who have followed the work of Mark Maneri, who studies nutrition in children with severe malnutrition in Africa and has the peanut butter-based formula. The studies of Dr. Gordon are done with Dr. Maneri in Malawi.
So really, what the CDI has done is it's helped us develop a network. And that network is studying basic biologic principles. What are the factors that are important in development of this microbial community? And then it's translating this new area of genomic medicine directly into pediatric clinical problems. looking at morbidities related to prematurity, looking at issues around antibiotic resistance. So with that, many thanks to the many collaborators that are present on the study. I want to point out Malik Nadow, our study coordinator, who really shares a lot of the success in terms of the study, particularly in terms of maintaining our family engagement. The pediatric gastroenterology group, Dr. Philip Tarr's laboratory and all the personnel that help with sample processing, the Center for Genome Sciences and System Biology, which we've talked about already, our biostatistical group, Dr. Bill Shannon, who was here earlier, and many of the community pediatricians and physicians who really provide us with the data that we need and take time out of their busy schedules to do that. And most importantly, the benefactors of the CDI, because really, without your all support, none of this really could be possible. So with that, I'll stop, and you can ask additional questions. You can ask more questions or feed your microbiome out there, so one of the two. Yes?
So many diseases are the result of inflammation. Mm-hmm. Are you thinking that all of this ultimately will be found coming from the gut?
No, and I don't. And it's like obesity. Clearly, it's much more complicated than that. But because the microbial community, those commensal bacteria, is now recognized to be so important in developing that balance of our immune system, that it's likely that it's an important component of it. Is it going to be the whole story? Not likely. But based on the most recent data, it's probably going to be, and it's likely to not be the same in each person. There's likely some genetic factors that will predispose you to having that kind of an effect, but won't predispose me to having that kind of an effect. And that's one of the reasons why we're also collecting DNA.
Mm-hmm. You mentioned, I know it was just an example of your daughter, how she might be only harvesting 500 of 1,000, and you're harvesting 1,000. Is that just an example, or is there really that much variation in the population?
No, I was exaggerating a little bit. But it is true, for example, what we do know based on the animal studies. So if you take mice that are raised on a Western diet, and you take their stool, okay, just the stool, and you inject that stool into a mouse that doesn't have any bacteria in it, and even if you restrict the calories of that second mouse with no bacteria, that second mouse will get fat, okay? So there's something about the bacteria that predisposes to obesity. How large and how significant an effect, I don't think we've really quantitated that yet. But there's a good 25 to 50% increase in terms of mouse fat. How that relates to humans is a little less clear. But that's kind of where I got that number from. It's a good question. Mm-hmm.
There are quite a few diseases that doctors don't know anything about yet that seem to have to do with antibiotics in your system, like IBS and things like that. Do you think these kind of studies will hold some answers?
Well, our hope really and our plan, because really to look at the disease states specifically, we'll have to, I mean, it's really, you're asking the ultimate question. We'll really have to follow this cohort out longer and longitudinally in time. And so, you know, the CDI has provided initial funding for this initial critical period, and we can look at some of these issues of growth within that period, which I think is really important and can pattern for later on. But it'll really take continuing to follow this cohort over time. You know, some people may have heard of the nurses study or the Framingham study where they follow them longitudinally. And I think to answer some of those questions, which are really the $10 million questions, it'll take a little bit longer time to do that.